Now, we're going to look at cell cycle, and then we're going to look at mitosis, which is part of the cell cycle. So what is a cell cycle? So it's a period where a cell is formed until a new cell, until two new daughter cells are formed. So from a time when a cell is formed until it divides into two daughter cells is called a complete cell cycle. Okay, so it first starts with, when a cell is formed, it will first start with interphase. Okay, interphase. And then 80% of the cell period is interphase. Okay, then after that is mitosis. Mitosis takes up 15% of the cell cycle time. Then followed by cytokinesis, which only take up 5% of the cell cycle time. Okay, so when you look at the cell under the microscope for plant cell, in the zone of cell division, you can either see the cell undergoing interphase, mitosis, or cytokinesis. So actually what happened to them during those phases, all right? Now, interphase is the longest phase of a cell cycle. So therefore, because it's the longest phase, 80% of the time, they are at interphase. So when you look at the zone of cell division, you will expect 80% of the cell will be at interphase, because this is by chance. If they take 80% of the time to be at interphase, that means when you look at the zone of cell division, by chance, 80% of the cells will be in interphase. So majority will be interphase. So what happened during interphase is the cell will grow, okay? And it will synthesize a lot of substances in the cell. Now I want to bring to attention that when we say the growth of organism, okay, is not the growth of cell. So when you refer to growth of organism, we say that the cell grow by, the organism grow by cell division and cell elongation, remember? Okay, so when you say, when the organism grow, it grows because the cell divides, okay, and then the cell enlarge. So you got more and more number of cells, and you got the cells enlarging as well. So that causes the organism to grow. But over here, we are talking about the growth of the cell. So we are referring to the cell when the cell grows. Actually, what happened when the cell grows is different from when the organism grows. So you have to be very clear about it. When you use the word growth for organism, it's different from growth for the cell. So in biology, this is where you need to pay attention to when you study biology. Grow with respect to what? Or growth with reference to what? So if you reference to different thing, it's different meaning. So these are the details you need to pay attention to. Okay, so during interphase is when we are referring to the cell. So the cell will grow. So grow means what? Grow means it will increase in size by synthesizing substances inside the cell. Okay, so interphase is divided further into three phases. Number one is G1 phase. G1 phase stands for gap 1. So G1 is gap 1 phase. Okay, and then number 2 is the synthesis phase. And then number 3 is the G2 phase. G2 phase is gap 2 phase. Now, why is it called gap phase? It's because when you look at the cells, we will find that cells during interphase look like nothing is happening. Okay, from the beginning to the end of interphase, the nucleus of the cells look the same. It's like nothing happening there. So it looks like it's a gap between mitosis. So that's why they call it gap phase. But actually, the cells are very actively growing. Okay, that's why it's called. That's why we call them. It's also a growth phase of the cell. So what do you mean by the cell grow? Cell grow means they must produce a lot of things inside the cell. So exactly what do they produce? First, they will produce a lot of mRNA, and then they'll use the mRNA to produce protein, Okay, and this protein can be structural protein of globular. So no, I don't say structural. So this is fibrous protein. You have learned two types of protein. Okay, so you're putting everything together. So fibrous protein or globular proteins. Okay, so fibrous proteins is to provide structural support to the cell. Okay, like microtubules, microfilaments. Okay, globular protein is functional protein. So this globular protein is the functional protein. Fibrous protein is the structural protein. Okay, so functional protein in the cell, you name it, enzymes, okay, carrier proteins. These are the proteins that they have learned. Channel proteins. Okay, what else? Antigens. Okay, so these are the proteins that you have learned that are functional. Receptor. Okay, glycoprotein. Okay, and then of course, using the enzymes, a lot other substances can be produced in the cell. Okay, because enzyme catalyzes metabolism in the cell. So anabolism may happen. New proteins are synthesized. New phospholipids are synthesized. Okay, new glycoproteins are synthesized. So therefore, the cells are considered as metabolically very active, and therefore mitochondria must be very actively producing a lot of ATP. So can you relate that? Based on what you have learned, you can actually list out all these things on your own. Okay, this is a revision. Okay, so this is actually the cells are actively growing and synthesizing substances, even though we cannot see it with our eyes. So that's why it's considered as gap phase. Okay, gap means like nothing's happening. It's a gap between mitosis. Okay, after gap 1 phase is S phase, is a synthesis phase. So at that time, DNA is being replicated, and then because DNA wrap around histone proteins, so histone proteins also must be synthesized for the new DNA to wrap around it. Okay, so after that is G2 phase, which is called the gap 2 phase. So again, G2 phase is same as G1 phase. It's a growth phase, continues. Okay, RNA synthesis, protein synthesis continue. And two additional things will happen, right? Centrioles will replicate to get ready for mitosis. So we know that we have got a pair of centrioles that is always arranged 90 degrees to each other. So when the centrioles replicate, you have another pair. So you've got two pairs already, getting ready for mitosis. Then organelles will also replicate, also getting ready for mitosis. For example, mitochondria and chloroplasts will also replicate so that they can divide evenly to the daughter cells after mitosis and cytokinesis. So this is a part where organelles and centrioles replicate. Okay, so gap 2 phase. So after gap 2 phase is mitosis, is only 15% of cell cycle. So you look at the zone of cell division, only 15% of the cells will be undergoing mitosis. Okay, so if you got 100 cells, around 15 cells will be undergoing mitosis, 80% of 80 cells will be interphase, during interphase. So mitosis is divided to PMET. Okay, so prophase, metaphase, anaphase, and telophase. So I have drawn it in such a way that the size of this section represents the length of the stage. So you can see that between PMET, right? Which stage is the shortest one? Curti.
Anaphase.
Yes, anaphase is the shortest one. That means to say, it's the fewest cell that you can see that's undergoing anaphase. Quite difficult to find one. Okay? All right. So last phase is cytokinesis. It's only 5% of the cell. So it's also quite difficult to find. Out of 100 cells that you see, only 5 will be undergoing cytokinesis. So cytokinesis means cytoplasm move to divide the cell into two. So cytoplasmic division. Okay. So we're going to look at, so this is the entire cell cycle. So every cell in our body, every cell in the plant would have gone through at least one cycle of this. It will have gone through G1 as G2 phase and then undergo mitosis. Okay, some cells after that will continue with the whole process of cell cycle. Some cell will then stop already. Okay, because they are already divided, they will then enlarge. So this phase here, interphase, is when they will enlarge. Okay, because a lot of synthesis happening in the cell. Okay, and they also take in water for plant cells. So this is when the cells grow in size. Not only they grow and synthesize things, they will also grow in size. So after that, some cells, a lot of cells will differentiate. So they will go into differentiation stage. So they will stop there. They will no longer undergo mitosis anymore for some cells. Okay, when they mature, they will not undergo mitosis already. Only cells in the zone of cell division will undergo mitosis. That means they will continue to divide. Okay, continue to divide. So cell cycle continue. Okay, but other mature cells, they will not divide anymore. Okay, so when they will not divide anymore, should I tell you or not? They will go into this stage called the G0 stage. Okay, there's this stage called G0, subscript 0. Okay, G0 stage means there's a gap stage where they forever remain in that. They will not change anymore. They will not undergo mitosis anymore. They will undergo cell division, cell elongation, and cell differentiation. And they remain there forever, like bone cell, okay? Nerve cell. Nerve cell will not divide anymore, okay? Okay, so let's go and look at mitosis. So we already said that when a cell divides, it undergoes two steps. Step 1 is mitosis, where the nucleus divides. Step 2 is cytokinesis, when the cytoplasm divides. Okay, so mitosis is divided into PMET stage. PMET is prophase, metaphase, anaphase, and telophase. So you want to memorize, you can use this one to memorize, but I already memorized it as PMET, so I don't have to use this to memorize. If you have not, you can use this plastic meat in tasty. So PMET, plastic meat in tasty. Okay, so visibly, how do they look like at this stage? Is present. Suddenly chromosome becomes visible, so it's present already. Then the chromosome arranged at the in the middle, so it's metaphase. Then the chromosomes start to move apart, so they separate. Then suddenly you got 2 nuclei in the cell, so it's 2, right? So present, middle, apart, 2. So this is exactly what you will see in the cell during these phases. And this is the actual meaning of those suffix being used there. Pro is before, okay? Before mitosis, at the beginning of mitosis. Meta is after. Ana is up or again, because anabolism is a building up process, so up, okay? Then telophase is the end phase. Remember I said telo is the end, so telomere is end of chromosome. So there's a very good advantage of you if you know the meaning of suffix of a biological word or prefix of a biological word, you will actually be able to work out the meaning of that word, okay? So based on the syllabus for mitosis, what exactly that they want you to learn is that you need to be able to describe the behavior of chromosome, nuclear envelope, cell surface membrane, and spindle fibers. Only these 4 things are during mitosis. So you can, this video is quite interesting. You can watch this, okay, when you revise the chapter. Today, if you have time, you will watch. If no time, you will not watch, okay? Okay, so these are the 3 cells that you need to draw into your notes. I have drawn the circle for you, so you need to fill in the blanks inside there to illustrate what happened to the cell during prophase. Okay? All right, so during prophase, now first you look at interphases. Now interphase look like a face of a person, right? Can you see that or not? And then you got the eyebrow and the little eyes there. So this is the 2 pairs of centrioles, okay? And then the nucleus is like a big mouth. and you go and look inside the mouth, you've got this mihun-like structure, okay? And then you got the lozenges there, okay? So that's how I remember lah, okay? So during interphase, you can see that the DNA are very loose, okay, loosely coiled around the histone protein. So it's a thin, loose thread-like structure, chromatin. So it's like mihun. Okay, so during prophase, what happened is that the chromatin will shorten and thicken by coil, loop coil, and then it will form the chromosome. So the mihun becomes mi. Okay. Then in animal got centrioles, so centrioles will move to opposite poles. So you can see that this is a pair of centriole, this is another pair. So this centriole here already moved to opposite poles. Then esters are formed. Now this line here is called the ester. Ester stands for star because it looks like the star. So esters are made up of microtubules that extend outwards or grow outwards from the centrioles. So it looks like a star. So it is called an ester. Okay. Then the nucleolus disappear. So the lozenges dissolve already. So nucleolus disappear. Okay. Then also the nuclear envelope disintegrates. So this nuclear envelope will break into small vesicles like this, and eventually it will disintegrate and disappear. So the mouth has disappeared, right? And then you can see what happened here is that spindle fibers, which is made of microtubules, they starts to extend from the centriole. Okay, you got the two poles, right? So spindle fibers, which is made of microtubules, so microtubules starts to extend from the poles, from both sides, both the poles, into the cell, okay, to form, each of this is called a spindle fiber. And why is it called spindle fiber? Because the entire structure is spindle shape. So this is called spindle shape. So that's why when the spindle fiber extend outwards like this, it makes a spindle shape. So it's called spindle fiber. Okay. Spindle shape is round at the center and sharp at the edge. So this is called spindle shape. Round at the center and sharp at the edge is called spindle. Okay. Okay, so now metaphase. Meta means middle, right? So you can see that all the chromosomes are arranged in the middle. So imagine if this is the Earth, and then the centrioles at opposite pole is the North Pole and South Pole. So the chromosome will be arranged at the equator. Equator is like the belt, okay, at the center circumference of the Earth. So this is the equator here, where the chromosomes will be arranged, okay, parallel, okay, to the line of the equator, perpendicular to the spindle fiber, okay, perpendicular means 90 degrees, okay. So chromosomes line up at equator of spindle fiber. You can see each chromosome is made up of two sister chromatids held at the centromere. So when the spindle fiber grows from the pole, so it will start to become longer and longer as it grows from the pole. And eventually it will attach to the centromere of the chromosome. So you attach at the centromere of the chromosome. Okay. So what is the role of centromere here? So now we learn that the role of centromere is to attach the two sister chromatids together, and also for the spindle fibers to attach to it during mitosis or meiosis. Okay, now anaphase. Anaphase, ana stands for apart. So the sister chromatids will separate apart, okay? So anaphase is the shortest phase, so therefore it's quite difficult to see. And the least cells are to be seen to be undergoing this stage. So step number 1 during anaphase is the centromere must divide into two, okay? Because centromere is a region where the sister chromatids attach to each other, right? So if this is my sister chromatids attached to each other, during anaphase, the centromere will have to divide. Divide means the sister chromatids will have to split already, because centromere is a region where the two chromatids are held together by certain bonds, like they're holding hands together. There are a lot of bonds that hold them together here. So during anaphase, they will have to divide. Divide means they no longer, the bonds that hold them together will break already. So then you can see two of them are still close to each other, but they are not holding hand anymore. So divide already. So when you divide, it becomes two, right? You can see it's two structure here now. It's not like one, okay? So centromeres divide into two. Take note of the word that you use. Divide, okay? Examiner will not allow students to use split. If you use the word split, you don't get any mark in exam. Okay, they divide into two. So then after that, spindle fiber will start to pull. You know, the spindle fiber will start to pull the sister chromatids apart. Okay. Now, what happens is like this. The moment, remember when I teach you, I say that this whole structure is considered as one chromosome, even though it is made up of two sister chromatids. Okay. Okay. So the reason why this is considered as a chromosome, I already explained, is because these two sister chromatids are attached together at the centromere. So therefore, this structure is considered as one structure only. Just like your body with two hands and two legs is one you. It's not two you, okay? It's one person, right? So therefore, everything is together. It's not separated into two yet, so it's called one chromosome, okay? So the moment, so when is it that we get two chromosomes is the moment the two sister chromatid divide, okay? Sorry, not two sister chromatid. The moment the centromere divide into two, now you can see that these two sister chromatids are two separate structure, right? So strictly speaking, when they separate into two separate structure, each of them is considered as a chromosome. So over here, you got two chromosomes. So that is when we call the two sister chromatid the two chromosomes. Okay, so when you learn with understanding, it's actually very easy to know when you call them sister chromatids, when you call them chromosome, when you call them one chromosome, when you call them two chromosomes. Okay, so the moment sister chromatids separate, we already call them chromosome, strictly speaking. So therefore, for me, I will label this, okay, as chromosome rather than sister chromatids. Okay, but some books will still label them as sister chromatids. Okay, it depends whether the standard of the book is a secondary school or A levels or degree. So the higher level you go, the more accurate and specific that you will learn. Because when you're learning at lower level, we will simplify things for you to learn. So we will not tell you the detail yet. So therefore, when it's simplified, sometimes it's not so accurate, but it's enough for that level for the student to learn. Okay, can you see that? So that's why when you go to university, you'll find it, hey, how come when I learn in A level, right, it's actually not this name, it's because it's being simplified, okay, to make it easy for you to learn. But when it's simplified, sometimes it's not so accurate, okay? So my sister chromatids will be pulled by spindle fiber to the opposite pole. So when the spindle fiber start to move, right, it moves by sliding effect. Okay, now telophase. Telophase means the end of, okay? So in telophase, what happened is you can see that telophase will start the moment when all the chromosomes already arrive at the opposite poles. So it starts when all chromosomes already finish separating. So then the chromosomes will uncoil and elongate to form chromatin. So the me will become mihun again. And the spindle fibers is no longer needed, so it will disintegrate. So in a cell, whatever that's no longer needed, it will be broken down. Okay, so that the subunit can be used to build other things in another part of the cell. Okay, so nuclear envelope reform. So the mouth appear again. Where's the mouth? Okay, the mouth appear again. Okay, then nucleoli reappear, so the lozenges appear again. Okay, so two daughter nuclei which are genetically identical. Genetically identical means got the same number and same type of chromosomes. They are formed. Okay, so this is telophase. Telophase only. Okay, now here this diagram here is actually showing you cytokinesis, which is not part of mitosis. Mitosis ends at telophase. Okay, so now we're going to look at cytokinesis in animal cell. What happens is that in animal cells, cytokinesis occurs by furrowing. Furrowing means narrowing of this part of the cell. Okay, if the membrane goes inwards, so this is called furrowing. Please learn up the word furrowing. Okay, so the membrane at the equator region starts to move inwards or narrow inwards, okay, until it cuts the cell into two. So how come the membrane can move inward? Is it will not move by itself. Something is pulling it, okay? So what is pulling it is the belt of microfilaments. Microfilaments are actually a protein which are fibrous, okay? So you got two strands of protein that twist and turn around itself very loosely. So this is the microfilaments, okay? So you got these microfilaments that arrange beneath the cell surface membrane inside the cell. So if this is the cell, okay, and I'm showing you the circumference of the equator. So you have got this belt, right, below the cell surface membrane inside the cell that will start to constrict inwards. So that means it will go inwards, like you pull the belt and make it smaller and smaller in its circumference, right? So as it moves smaller and smaller like this, and also the membrane is attached to it, so the membrane will also be pulled inwards. So that's why the membrane furrow or narrow inwards, okay? Because of this microfilaments beneath the cell surface membrane that constrict inwards, pulling the membrane to go in, okay? So organelles are eventually distributed into the two daughter cells. So now we already get two daughter cells. So you need to draw all those. Now for plant cell is different. Plant cell cannot furrow because the cell wall is rigid. Okay. So for plant cell during cytokinesis, what happened is that a cell plate is formed in the center of the cell at the equator region. All right. So this cell plate is formed from the fluid-filled vesicles which come from Golgi apparatus. Remember, Golgi apparatus is an organelle that will collect, modify, transport, and distribute substances. Okay. Center of collection, modification, transportation, and distribution. So after having produced the cell wall is made out of cellulose, hemicellulose, and pectin. So after having produced hemicellulose, cellulose, and pectin, okay, modify already in the Golgi apparatus, it will butt off to become vesicles. So the vesicle move to from the Golgi apparatus to the equator region of the cell, and slowly, these vesicles will fuse together, elongate all the way until it touches the parental cell wall, the old cell wall, okay? And therefore divide the cell into two. So this is called the cell plate, okay? The cell plate at the center of the cell at the equator region. So cellulose in vesicles will form the new cellulose cell wall. Pectin in the vesicles will form the middle lamella. Remember middle lamella? The middle plate is like a cement that join two neighboring cell walls together. So it sits right in the center of the cell wall of two neighboring cells because it cements the two cell wall together. Okay, then the membrane of the vesicles will form the new cell surface membrane. So the cell surface membrane here, okay, this cell surface membrane actually comes from the membrane of the vesicles, which comes from the Golgi apparatus. Okay, so this is an animation made especially by our another teacher, Mr. Vilas Agas, because he loves to make animation and he's very generously sharing with other teachers. Okay, so step 1. So this showing you the prophase, right? Where centrioles move to opposite pole, nuclear membrane disintegrate, chromatin becomes chromosome, and which is made up of two sister chromatids held at the centromere. Then, okay, then all the chromosomes align at the equator. Spindle fiber extend from the centriole and then attaches to the centromere of the chromosome. During anaphase, the sister chromatids, the centromere divide, sister chromatids are pulled apart to opposite pole by spindle fibers by the sliding effect. Okay, so please pay attention when the sister chromatids are pulled apart, right? Sometimes students are asked to draw in exam one. You must draw V shape. Can you see that this is actually V shape because of the force that pulls it? So when there's a force that pulls it here, then because the pulling force is here, right? So the rest will all be delayed. So therefore it becomes V shape. So you have to draw V shape. Okay. So what is this thing here? Is actually the centromere that is drawing here. Okay, the centromere. So after that, when all the chromosomes arrive at the centrioles, then it's already telophase where the nuclear membrane reform, nucleolus reform. Then chromosome will uncoil and elongate, become chromatin. Okay, so after that, the cytokinesis will happen, right? So I think, can you see this black color region here? This black color region shows you cytokinesis. Cytokinesis can overlap with telophase one. Sometimes cytokinesis can overlap with telophase. Okay. So here, this is a diagram to show you the different stages of mitosis. And interestingly, they say that this is in a plant cell. Okay. But what happens is that we cannot see the cell wall because the slide has been processed in a way where the cell wall are digested away. So there's no more cell wall there, okay, because it already digested away. So what you can see here is interphase. You cannot see much things, okay, because all the DNA are in the form of chromatin, thin thread-like structure. Then you look at the prophase. See how prophase is different from interphase. You can see that because the chromatin start to condense and thicken, a lot of empty spaces starts to be visible. So this is something that you want to be able to identify. There are no specific arrangement of chromosome yet, but you can see a lot of spaces starts to form because the chromatin start to condense and thicken. So it will leave behind some spaces, irregularly, randomly arranged spaces. So that is the typical structure of prophase. Okay. Now this is still prophase. This is still prophase. Okay. Metaphase is when you can see all the chromosomes are aligned at the equator, is metaphase. Right. The red color line are the spindle fibers. Now anaphase is when you can see V-shaped chromosome, and you've got two of them, both moving to opposite poles. Okay. Then after that, when you see that two black structures at the end, at the two ends, is actually the telophase already. Okay. Over here, can you see that this is the cell plate that's formed at the center of the plant cell? So because it's a plant cell, there's no cytokinesis, but it's actually, there is cytokinesis, but cytokinesis is not by furrowing. Okay, it's by formation of cell plate at the equator region. Okay. So let us look at more diagrams so that you know how to identify the structures, okay? Now, this is interphase. Now, sometimes in interphase, you can see, don't wanna, okay, where's my arrow? Hang on. So I'm using arrow to point. So interphase, sometimes you can see the nucleolus one, can you see that? Okay. And there's no specific pattern. You can see there's no specific pattern. And the nucleus appear as a very huge, circle inside the center of the cell. Okay. So, and the cell is almost square. Usually it's square one, okay, but this cell is not very square. Usually it's square. So the nucleus will look very big at the center of the cell. Now look at this structure here. This structure is different from this one. This structure, can see a lot of spaces are starts to form. So this is actually prophase already. Very easy to identify, okay? Why is it my name is there? Okay. Undo, cannot undo. What happened? Okay, I don't know what happened. Okay, so, all right. So I'm pointing it, okay? So next. So can you see prophase? A lot of empty spaces starts to be formed because chromatin condense. Nucleolus is still there. You can still see it, okay? Nucleus is right at the center of the cell. It looks very big. Nucleus looks very big, okay? Now, again, can you see that? Now I can see it's a later stage of prophase where you can see chromosomes already, but there's no specific arrangement pattern. So it's still prophase. This is metaphase already because all the chromosomes are in the center of the cell, right? So this is called the equator. So when you describe, you need to say that the chromosomes are lined up at the equator of the cell, okay? So here you can see that the chromosomes are separated to opposite poles already. They are being pulled to opposite pole, and you can see the V shape. So this is Anaphase. Then finally, you can see in one cell, you got two structures here, but you cannot see the V shape anymore. So this is Telophase because the chromosomes already finished separating, already arrived to the pole already. So therefore, you cannot see the V shape. Now, I want you to also pay attention to this cell. These two cells, I don't like to use red color. Okay, look at the arrow, these two cells. Can you see that the size of these two cells, each of the cell is actually almost half the size of this cell? Okay, because after the cell divide, right, when the cell plate is formed at the center, the new cell that is formed, the daughter cell will be of this size already. Can you see it becomes square again? Okay, it becomes square again. Right? So you will be able to, based on the size of the cell, you will know that these two cells are newly formed. Okay, these two cells are newly formed because the size of this cell is half of this neighboring cell. So these two cells are newly formed, and this is the new cell plate. Okay. The new cell plate that extend outwards to form the new cell wall. So it's very easy to identify based on the pattern, based on the size, okay, and location. So this is an actual micrograph that is taken in our lab, okay, using our microscope during mitosis practical, okay, using a root tip slide. So root tip, remember, I explained to you before, in the lab, in order to view mitosis in the root tip, we will grow the roots from the onion, right? We will put the onion, the bottom part of onion to the water, touching the water to stimulate the root to start to grow. So after that, we take the root to make the slides, okay? So you can see here, this one is prophase. You can see not? This prophase, because why? Because you can see empty spaces there. The empty spaces are formed because the chromatin start to condense and thicken. So this one is interphase. Can you see not? This one looks like telophase. Can you see not? This metaphase here. This is like metaphase here. Okay. All right. This is anaphase. Can you see very few cells undergoing anaphase because the shortest phase. So this is anaphase. Okay. This is definitely interphase. These two cells could be two newly formed cells. Okay. So these are the different phases in the cell. Now this one here is animal cell undergoing mitosis. So this is prophase because chromosomes are visible already, but no specific pattern. This metaphase, chromosome arranged at equator, okay, parallel to the equator, but perpendicular to spindle fiber. Okay, you can see that. Then V shape of chromosomes, so anaphase, still anaphase because still V shape. Then one cannot see V shape already. So this telophase. This one is we look at the tissue in the body. Inside the tissue body, there'll be some cells that will undergo mitosis. Like in this case, you can see this is lymphoid tissue of our body. You can see this is cell undergoing metaphase, and this one looks like telophase. Can you see that? Okay, and the rest of them are actually interphase. Okay, interphase. This one, you can see this cell here, very interesting. This cell got two nucleoli, right? Because the nucleus of the cell may contain one nucleolus or more than one nucleoli one. So that's why you can see here got two nucleoli, which is actually quite normal. Okay. Now here, this is undergoing anaphase, V-shape anaphase. Okay. So this is in cervix epithelial cell. Now, epithelial cells are like our skin cells, right? Which align the surface of our body which faces external environment. So it can be on your skin, it can be your airways, like trachea, bronchus, bronchiole, alveoli. It can be your intestine, urinary tract. All the cells that line those tubes are called the epithelial cell. Epi means on the surface of your body facing external environment. Like cervix also is facing external environment one. Okay, because you follow the tube, you can see sunlight. That means it's connected to external environment. So external environment is considered as external environment. Okay, so this kind of cells, right, will always shed away, and therefore we have to keep on regenerating the cell by cell division. So mitosis is part of cell division. So you can see the cells undergoing mitosis here. Okay, for regeneration, for replacing the old cells. Okay, replacing the old cells. Okay, now duodenum is in our digestive tract, right? So again, the epithelial cells of duodenum also will shed, and therefore continuously there will be mitosis to replace the cell. Okay, so here this is metaphase. Metaphase is very easy to identify. This is metaphase again. Okay, this is telophase. This telophase. Okay, so homework is you need to draw all the cells undergoing mitosis just now when I explain, okay, all the cells into your notes and label them. And also you need to draw all this. Imagine these are the micrograph given in exam time, and you are asked to draw this.